Yeah, thanks for the introduction. I want to thank the organizers for putting together a wonderful symposium. I've really enjoyed the lineup of speakers that we've had, and thank you guys for your time to come out. So I'm a professor of mechanical engineering, so I'm not a cell biologist, but I work in this area for cell biomechanics. These are my disclosures. So for me, I'm interested in cells as athletes, and there's been some iconic videos that we've seen today as well as from the past of cells migrating, doing amazing feats of strength, and they use their forces to be able to regulate their function. They use this in normal ways for health, and these processes go awry in diseases. And so cells have their ability to contract as one of their fundamental properties for migration. If you're talking about cardiomyocytes, their ability to actually contract the heart and eject blood. They use it for cell adhesion, like in the case of platelets that are gripping tight to a wound site. Or if you have groups of cells, whether it be clusters of cells, or I'll talk about today, endothelial cells, you have junctional barriers or cell contact that's maintained by tugging forces between these cells. And as a mechanical engineer, I'm very interested in the machinery of cells. And so there's been some wonderful imaging of the cytoskeleton in action as a cell crawls around and migrates. You can make out the individual stress fibers that are being formed, the focal adhesions where the cell has attachments to its surface. And so you can work out this system behavior going on within the cells. And so I look at this in terms of a mechanical paradigm. You have polymerizing filaments, you have motor proteins, and you have attachment points like the integrins and cadherins that allow the cell to grip and move. Now, to measure these forces, you have to go beyond microscopy. You can watch this process happening, but really you want to know values. You want to know forces, how much, where is it being directed, what are the dynamics of this process. And so my lab has been using a lot of microfabrication technology. These are silicone micropillars or microposts that you can grow cells on. So a cell would be attached to the top of these pillars, which are around 3 micrometers in diameter and 10 micrometers tall. And the cell spans across multiple ones. And since this is a silicone rubber, these are very flexible. And so as a cell crawls, it begins to pull and tug on these cantilevers. And now, just through some simple equations, you can actually measure how much force is being generated by these cells by tracking the displacement of those tips. So using just optical microscopy or fluorescent microscopy, we can actually track the position of these pillars and using that as a calculation for how much force there was based upon the dimensions of these pillars as well as the mechanical properties of that silicone. So these are very small structures that we make in the clean room at first. We normally take the same technology that you use to make your semiconductor chips. This is photolithography, which we all benefit from with our smartphones. But you can use this now to create three-dimensional structures. So you can start with a silicon wafer that you can put on a photosensitive polymer, and wherever light shines on that polymer will cause it to become cross-linked. And then you can wash away the uncross-linked material, leaving behind your structure that you have. Now, this takes some time and effort, so we want to be able to replicate this over and over again. So we do a three-dimensional photocopy where we actually pour that PDMS across our structures to create a mold. So you can think of this like a cupcake tray. And then from this, we can then pour in our silicone a second time to actually get our structures. And so these are arrays. What you're looking at here is each square has 100,000 of those little pillars looking back at you. And so this has been fabricated on a glass cover slip that you can drop into your 6-well dishes or do live microscopy with. Now, to get the cells to stick, we have to use microcontact printing. And if any of you ever have used rubber stamps when you were kids, it's a very similar process. You ink the stamp and then make contact with your surface to transfer your solution. In this case, our solution is our extracellular matrix. So we would start with maybe fibronectin in solution or laminin in solution. And what happens is because PDMS is hydrophobic, you get absorption of that protein onto the surface. So when you bring your stamp into contact with your posts, wherever you have contact, you get protein transfer onto the tips. And so depending upon the shape of your stamp, you could predict, or I should say control, which posts get that stamp and which do not, which ones have protein and which do not. But wherever you did not stamp, you can then coat it with a non-adhesive to prevent any additional proteins from sticking. So one of the first studies that we did was we started looking at the role of cell shape as well as stiffness. So what you're seeing over here are HPECs, endothelial cells, that have been stamped to control their cell area. So you have small cells and large cells, or I would say cells that were allowed to spread out and cells that were not. And we also did these on pillars of different heights, so that way you could control the stiffness of these structures. And what we found here was that by increasing the spread area, you're able to increase the amount of force that these cells generate. At the same time, too, you can also increase the stiffness of the environment to also increase the forces. And likewise, where you see these forces being applied, you're also seeing larger focal adhesion areas at those positions. Now, going further, we've actually used these stamps to create lines and to analyze cell migration, not in a three-dimensional content, not in a two-dimensional tissue culture dish, but in a one-dimensional environment, which is sort of like cells maybe crawling across a collagen fiber. So what we've done here is we've stamped lines of matrix and allowed a cell to spread out. And we started watching the dynamics of these forces as it did this. So as a cell is migrating from to your right, it reaches out and starts to attach to the next post. And as it begins to pull and tug, we can actually measure how much force there is. But there's an interesting dynamic. And typically in cell migration, you think about extension, new adhesion, contraction, and then release. But what we weren't seeing was really coordination between the activity at the front of a cell and the release at the back end. So if you kind of look at these in a time lapse here, there's a local interaction where the cell forms a new adhesion, begins to contract, and at the same time lets go of the previous post that was at the front end. And then when it forms its next attachment and begins to contract, this one lets go. But you don't really see any coordinated activity going on in the back end. So there's almost like Slinky the dog from Toy Story, where it's walking along in the front end and the back end just kind of follows along afterwards. Now, cell migration is pretty important for development, or I say growth formation of the skull. And so one area we've looked at with our collaborators at Seattle Children's is in craniosynostosis. This is in collaboration with Michael Cunningham there, where he's collected a bunch of patient samples from individuals who've had premature suture closure, and also some control samples as well. So with these osteoblasts, what Michael has seen was that there's a high level of IGF-1 expression in individuals who have craniosynostosis. And what we found was that this high level IGF-1 expression affects the ability, or actually increases the contractility in these cells and reduces their migration. So what you're seeing over here are some control osteoblasts and some of these patient-derived craniosynostosis osteoblasts. And you're seeing a lot less migration within these cells and a lot higher migration over here. And basically by looking at these forces and migrations, we can either add some exogenous IGF-1 or inhibit the receptor or affect the contractility with blebbistatin to tailor or control this role of IGF-1 on migration and on forces. Now, shifting gears, I want to talk about cardiomyocytes. So with Chuck Murray and collaborators at the UW Medicine, there's a large effort towards taking stem cell or pluripotent stem cells and differentiating them into cardiomyocytes. And these cells, they beat, they contract, but the question really is how are they the right kinds of athletes that you would want to be able to put back into an infarcted region? And so what we've been working on is actually measuring these forces of these new cardiomyocytes. So this is a stem cell-derived cardiomyocyte contracting on our micropost, and you can see each twitch contraction happening within the cell. We can electrically pace these cells to also control the frequency of this response. Who's at the door? We've also been working as a kind of a biofunctional assay. We have over here a thyroid hormone that helps to increase these twitch forces, or working with Hanella Ruhala-Baker, we've actually been working with some microRNA that can help the maturation process of these cardiomyocytes. Now, going into 3D, we've been working on these tissue-engineered constructs using these stem cell-derived cardiomyocytes. And instead of using microposts, we're using these much larger silicone posts. So what you're seeing here are engineered heart tissue, or what I like to call heart strings. And so if, these heart strings are pulling on these silicone posts, where one of them has a glass rod inserted inside of it, and the other one has a magnetic, has a rare earth magnet embedded in that post. And what's nice about these three-dimensional constructs is that these cells have beautiful sarcomeres within them. They have a much more elongated shape that you'd expect to see from tissue, and they function as a complete tissue because if you watch them under a dish, they beat. So each of these heart strings are beating to their own different drummer, but you can track that motion with the movement of this post that has the magnet within it. Now, we've developed some optical techniques to analyze all of these tissues at once in a 24-well format, but we've also done this magnetically, where we've used the movement of that magnet in the tip with a magnetometer to measure that change in magnetic field. So we have these GMR, giant magnetoresistive sensors underneath here that can detect the change in the magnetic field as these posts are being bent by those heart tissues. So we've been able to do this for real-time measurements of this contractile function as we tried out different drugs in a 24-well dish. Changing gears one more time, we've worked a lot on platelets, which are some of the smallest cells of our body. Platelets here are involved in the clotting process. They help to grip onto a wound site. They help in the fibrin formation, and they also help in clot retraction. And these platelets are often found clustered together in tight packs, which help to maintain the clotting potential. They are compacted together. It prevents enzymes from breaking apart the clot. It pulls everything into a tight, compact form. And so to measure these forces, we've been using the Micropost, where we have groups of these platelets seated onto these pillars over here, and we can then measure their forces. And we can try out different drugs that inhibit the force generation. Well, first of all, we can try thrombin out, which increases the contractility, but we can use Y27 or ML7 or blebbistatin to relax these cells and cause an inhibition of their contraction. Now, if you want to look at individual platelets, these pillars are rather big. You can see right here a platelet or two hanging out on the tips of one of these pillars. So we've gone nano. We've actually created these silicone nanoposts that are around 800 nanometers in diameter. And what you're seeing here is an individual platelet that's normally around 2 to 4 micrometers in diameter that has been spread out across the tips of these pillars and generating force. And so in collaboration with Jose Lopez at Bloodworks Northwest, we've been looking at the different receptors that are involved in this force transmission of the cytoskeletal tension from a platelet to the outside world. And now we've also done this in a microfluidic environment where we can have blood flowing through these microchannels, where we can actually have a block and post structure that's built up as an array within this channel. And over time, the platelets will aggregate and form a clump. And you can actually watch that consolidation, that contraction process over time as it causes this micropost to bend. And what's happening is the flow, as the platelets are traveling across this block and post, are experiencing a high fluid shear that's activating them. So if you look at these structures, we have these aggregates that are forming on the blocks and posts. We do get some that are bound to the walls or bottom of the channel, but these are large masses of platelets that are engulfing those block and posts. But if we looked earlier on in time, right when these platelets first started to attach, you see that there's quite just a few number of platelets that are able to attach, or that are generating force. This initial connection is just 1 or 2 platelets, and then eventually more build up. This one right here is pretty strong. You should probably put a red cape on him because of his Superman abilities. But we can look at these forces and see when you have normal forces being generated by a patient, or if there might be on aspirin, you have an inhibition of this force generation. So this is a way of looking at different antiplatelet medications or looking at diseases that might arise in a patient due to trauma-induced coagulopathy. And the last story I wanted to share was our work on endothelial cells. So these are the cells that line the walls of our blood vessels. And they have a relationship with each other that's mechanical. They have tugging forces that they generate with each other to help maintain their barriers. So we can use our microposts, and normally you would see a cell on 2 posts, or sorry, 1 cell on posts, or if you had 2 cells on posts, they actually create a junction between them, and you can see that the sum of the forces all equals 0. All of these posts are bending towards a middle point here. And we can use that to calculate how much force is being generated between these pairs of cells. So we can stamp these bow ties and allow the cells to attach and look at beta-catenin over here. And we can see that where we have large junctions, we have large tugging forces between the cells. But over here, we have small junctions and small tugging forces. So by using different manipulations, we can dial up or dial down these tugging forces and see a relationship here with the junctional size. So you have a trend where as you increase the tugging force between the cells, you see an increase in that junctional size. Now, in a fluid environment, these cells are being subjected to laminar and disturbed flow. And so you can recapitulate that by using a flow chamber where you can use this backward-facing step to create regions of disturbance and regions of laminar flow. So this might be the flow that you would see in a straight blood vessel, but back here is the recirculation that you might see where you have a bifurcation in a blood vessel or behind a valve. And these are regions where you tend to see more endothelial dysfunction. And so we can look at the biomechanics of these cells in these 2 different kinds of flow regimes. And so what we found here was that under that laminar flow, these cells have higher intercellular forces or tugging forces. You can see over here a monolayer. We can calculate what's the net force acting on one of these cells. While under these disturbed flows, you see lower forces. And this correlates with the kinds of junctions. We see increased beta-catenin length with laminar flow, as same thing with ZO1 too. So now what you're seeing here is that the cells are using their forces to maintain the tissue, but they're also sensitive to the external forces that are acting on them. And so you have this kind of force response. The last thing I wanted to show here was just something that we've been developing now where instead of using microposts, we're using soft PDMS. So this is a much softer version of PDMS that's more commonly used. This is around 5 kilopascals. And what we can do is we can stamp a pattern of black dots on this. And then we can grow our cardiomyocytes on it. And because it's a much softer environment, let's see if we can do it this way. There we go. There we go. What you're watching here is the alpha-actinin within these cells. And because you have a much more compliant environment, you're able to see the dynamic contraction of all these sarcomeres here. And it's rather interesting to us that in these immature cells, it's not fully synchronized. You have some regions that begin to contract early on and some that follow afterwards. But with this, we can now measure out the forces. Let me see if I can play this real quick here. There we go. So we can actually track the movement of those black dots in our green field. And this time we just kind of reverse the image around. But we can actually track those forces being generated. So what we're hoping to do is to actually correlate those internal movement that you see in the sarcomeres with the external forces that we see in our that silicone substrate. So I showed you guys some different tools that we developed. We're using these flexible microposts to measure cell forces. We can also control the cell shape with microcontact printing, and we're interested in how mechanical cues like flow or stiffness or stretch, how are these affecting the behavior of our cells of interest? And so we've used this for cell migration to look at how cells coordinate their traction forces, or looking at cardiomyocyte contraction. Also looked at the role of platelets, whether it be single platelets or a group of platelets when they're forming a clot, and also showed a lot of work about the junctional integrity that's used in endothelial cells and how the mechanobiology of their environment plays a role there. So I'd like to just thank the trainees in my lab, as well as my collaborators here in Seattle and elsewhere, as well as thanking the funding agencies. Thank you.
We have time for a couple of questions.
You were hinting at the platelet function assay of potentially having clinical laboratory value. Is that something that you're looking into, and do you think this can be going above and beyond what we can currently do with platelet aggregation testing in the clinical laboratory?
Yeah, when you're thinking about like platelet function assays, there's been force assays in the past. You know, in the late 70s, there was these clot strips. But a lot of the platelet function assays now are mostly about, are platelets becoming activated? Do they go through a shape change, or do they stick? And so actually that's what we've been working on, is using the force measurement as a functional assay for platelets. So we've just wrapped up an observational trial at Harborview where we've actually looked at trauma patients that have come in. And what we found was that we measured the blood of those patients upon admission to the emergency department, and then we tracked their outcomes. And what we found was the patients that needed transfusions, these are the patients that probably had the most bleeding, those that required platelet transfusions, had lower platelet forces upon admission to the emergency department. So in one hope is that this type of technology could be used as a quick assay to measure whether a person's platelets are functional or not, to help guide their treatment. And that's actually part of the spin-out company, Stasis Medical, from my lab.
So I'm just curious about the cardiomyocyte work. Do you envision that the mini-pills that you have used, can you use it for maturation of the cells before transplanting? And have you also tried some of the cardiomyopathy samples, the cardiomyopathy IPS-driven from cardiomyopathy patients for this type of work?
Yeah, so we have a collaboration with some colleagues in University of Colorado, Denver, looking at Danon disease from iPS-derived patient samples. There we're looking to see the, kind of trying to understand what's wrong with the contractile machinery within these patient-derived cardiomyocytes. We do quite a lot with changing the stiffness to increase the maturation of the cardiomyocytes, but where we're really using them a lot is in terms of like a functional readout. So as we try to develop better protocols for maturing these cardiomyocytes, you may look like a cardiomyocyte, you may have the structures, but it'd be really nice to see if you can do the work of a cardiomyocyte, and that's really where the micropillar allow us to sort of measure out that the performance of these cells.